In today's lecture, we will be covering bioinorganic chemistry, metals, and medicine. We will start off doing a very brief review of deoxyribonucleic acid, or DNA, as this will be needed to understand how many chemotherapy agents work. As you may already know, DNA contains all of the genetic information for a cell. What that means is that DNA contains all of the information that the cell needs to function properly. For example, all of the proteins and enzymes that we have already talked about in this course have their polypeptide sequences encoded in DNA. Therefore, without DNA, cells would be unable to make any of those proteins. Another way to think of it is that DNA contains the ultimate blueprints for almost all cell structures. So DNA is vitally important, and any significant disruptions to DNA can have severe consequences. When looking at the chemical structure of DNA molecules, they are long polymer chains made up of alternating trimers of deoxyribose sugar, phosphate, and a nitrogen-containing base. The word trimers simply refers to the fact that the repeating units of DNA have three distinct parts that make them up, as shown here. So this would be the trimer portion, where you can see the deoxyribose sugar here, the phosphate here, and this would be the nitrogen-containing base. And all of these together make up the repeating trimer unit. So this is one trimer, this is another trimer, etc., etc. DNA polymers, or strands as they are often referred to, generally do not exist as isolated strands, and are instead hydrogen bonded to another complementary strand. These two strands form a double helix type structure, as shown right here, with the blue ribbon representing the sugar and phosphate backbone. The two DNA strands are anti-parallel in that one strand will be oriented with the phosphate and sugar backbone pointed one way, while the complementary strand has the phosphate and sugar backbone oriented the other way. So one would have the phosphates and sugars facing this way, and then the complementary strand would have them facing the other way. But what do I mean by complementary DNA strands? And what are the nitrogen-containing bases in DNA? Well, the nitrogen-containing bases are often referred to as base pairs, and there are four main bases in DNA. Thymine, often abbreviated as T, adenine, often abbreviated as A, cytosine, often abbreviated as C, and guanine, often abbreviated as G. Adenine and guanine are referred to as purines, while cytosine and thymine are referred to as pyrimidines. The reason they are called base pairs is because they preferentially hydrogen bond to only one of the other bases, or the proper complementary base. As shown right here, thymine and adenine are complementary, and cytosine and guanine are complementary. Notice thymine and adenine preferentially form two hydrogen bonds, so right here there's only two, while cytosine and guanine preferentially form three hydrogen bonds with each other, as shown right there. So looking at a DNA double helix, we can see that the two DNA strands are not identical, but contain complementary base pairs that hydrogen bond together. So that's represented here, where you can see these base pairs hydrogen bonded together. Additionally, the nitrogen-containing bases are aromatic in nature, so they are fairly planar. As such, the base pairs are stacked in parallel planes on the inner portion of the DNA double helix. So you can see these are going to be stacked parallel to each other. During cell mitosis, when a cell replicates and splits into two cells, the DNA helix unzips and each single strand of DNA is used as a template to generate a new complementary strand of DNA. The two identical DNA double helices then separate into two new cells, where one of the strands in each of the two new cells came from the original cell that split. The enzyme DNA polymerase is the protein that forms the new DNA strands using one of the original strands as a template. DNA polymerase does not copy the template strand, but forms a complementary DNA strand with complementary bases. Now we will move on to transition metal complexes that are used for medical applications. We will start with cisplatin, or cis-diamine dichloroplatinaum 2, which we have encountered several times in this course already. You can see the structure of cisplatin right here. Cisplatin is a very potent chemotherapy agent, and it works by disrupting DNA replication. This causes cell apoptosis, or programmed cell death, during mitosis. Why would we inject people with what is essentially a poison that kills cells during replication? How is this going to help a cancer patient? Well, the reason cisplatin and many other chemotherapy agents work is that cancer cells replicate much more quickly and much more often than normal cells. This uncontrolled replication is what causes tumor formation. If you have a chemical that kills cells specifically during mitosis, then it will disproportionately affect cancer cells, which replicate much more quickly and much more often than normal cells. You essentially count on the fact that you poison the cancer cells faster than most of your normal cells, and you control the dosing such that the normal cells are affected as little as possible. However, because cisplatin kills normal cells too, there are quite a few side effects that are seen with cisplatin treatments. The side effects are disproportionately seen in cells that are fast replicating under normal conditions. This includes hair follicles, skin cells, and cells lining the gastrointestinal tract. This also explains why many cancer patients undergoing chemotherapy experience hair loss, rashes, and gastrointestinal distress. So we know that cisplatin is a good chemotherapy agent. But how does such a simple transition metal complex kill cells? First off, cisplatin is injected intravenously into the bloodstream where its structure remains unchanged. The chloride ligands are somewhat prone to substitution, or are somewhat labile, and you should be able to explain why using the trans effect, but in the bloodstream there is a relatively high concentration of chloride anions. This prevents chloride substitution according to Le Chatelier's principle. This keeps the cisplatin neutral, which helps with diffusing across the phospholipid bilayer of cells. Inside of the cell, however, the chloride ligands are substituted because the concentration of chloride anions is much lower. Once again, you should be able to justify why the chloride ligands are prone to substitution, and you should be able to identify the likely mechanism of substitution. The reaction shown right here is likely to go through an associative ligand substitution reaction because the platinum complex is a 16-electron species and coordinatively unsaturated with small ligands. The cis-diamine diaquoplatinum 2 that forms inside of cells is actually the active species that causes cell death. Water ligands in cis-diamine diaquoplatinum 2 are substitutionally labile. And when the platinum complex enters the cell nucleus, it will bind to DNA. Specifically, the platinum will bind to two guanine bases that are in close proximity through this nitrogen highlighted here, which is not involved in hydrogen bonding. The platinum species could also potentially bind to adenine, which also has a nitrogen that isn't involved in hydrogen bonding, but it has been determined that it preferentially binds guanine. When the platinum complex binds to DNA in this way, as shown in the picture to the right, it causes a bend or a kink in the DNA double helix. This kink prevents proper DNA replication during cell mitosis. Additionally, The platinum binding is resistant to DNA repair mechanisms present in the cell, which can cut out sections of DNA, say if there were mismatched base pairs, and replace them with proper ones. Based on this mechanism, can you explain why trans-diamine dichloroplatinum 2 is not an active chemotherapy agent? The cis binding mode forms the necessary kink to disrupt cell function, prevent DNA replication, and cause cell death or apoptosis. A trans-binding mode, on the other hand, could potentially link two DNA strands together, but wouldn't cause a kink in the double helix, and would not disrupt the DNA structure enough to prevent DNA replication and cause cell death. So this is why it needs to be cis-diamine dichloroplatinum 2. A major issue with cisplatin, however, as we mentioned previously, was that it has many side effects that cause cancer patients significant pain. Many of these issues are caused by the lability of the chloride ligands, which cause the cisplatin to leave the bloodstream relatively quickly and gives less time for the metal complex to reach cancerous cells. Furthermore, there are types of cancer that become more and more resistant to cisplatin treatments as the cancerous cells develop mechanisms to repair the damage caused by cisplatin. Because of this, there has been a lot of research that has gone into the area of designing platinum-based complexes for chemotherapy applications. One such example is carboplatin, shown to the right here, which has a bidentate ligand replacing the chloride ligands. The bidentate ligand is much less labile than the chloride ligands, and you should be able to explain why using the knowledge you have learned in this course. Because of the less labile bidentate ligand, carboplatin stays in the bloodstream longer, which allows more time for the drug to reach cancerous cells. This also causes fewer side effects. However, because the bidentate ligand is less labile, carboplatin is less toxic in general and less potent versus cancer cells. This metal complex therefore needs to be administered in higher doses than cisplatin and is often used in conjunction with other chemotherapy agents. Another example shown below of another type of platinum-based chemotherapy agent is oxaliplatin, which is more effective against cisplatin-resistant cancers. There are numerous other examples, but most platinum-based chemotherapy agents have similar square planar structures. Different ligands are used to give a variety of properties that are desirable. For example, picoplatin, shown here, can be administered orally. Most of the platinum-based drugs that have been developed are not FDA approved yet, though, and there are a large number that are still undergoing trials. At this point, we're going to switch gears a bit and take a brief look at metal complexes that are used for medical imaging. We will first talk about magnetic resonance imaging, or MRI, contrast agents. MRI is essentially the same thing as NMR, except it is in 3 dimensions and used for people. Most MRI contrast agents are gadolinium-based complexes, and they work by allowing the nuclei to relax more quickly. MRI contrast agents make use of multidentate ligands to prevent the release of free gadolinium into the bloodstream. This is important because free gadolinium is excreted through urine. And for patients with impaired kidney function, this can cause serious and potentially life-threatening complications. One popular MRI contrast agent is Magnavist, shown above, which has an octadentate ligand. It, however, cannot be used with patients that have impaired kidney function. Gatavist, on the other hand, shown above to the right, also makes use of an octadentate ligand, but it can be used with patients with impaired kidney function. Can you explain why there would be this difference between Magnavist and Gatavist based on your previous knowledge from this course? They both have similar denticities, but Gatavist employs a macrocyclic ligand, so it is less substitutionally labile for entropic reasons. Next, we will take a brief look at radiopharmaceuticals, which are used for nuclear medicine imaging. In particular, we will look at technetium-99m, which is pretty much the only transition metal where all of its isotopes are radioactive. In addition, technetium-99m is the lightest element where all of its isotopes are radioactive. Technetium-99m is specifically used because it has a half-life of 6 hours. This half-life is in a bit of a sweet spot because it is not too short, so you have time to prepare and ship the imaging agent, but not too long so that it stays radioactive for a very long time. The way this type of imaging works is that the radioactive decay of technetium-99m can be followed in the body to visualize various organs. For example, Colatex, shown to the right, is used to image liver uptake and gallbladder/bile duct function. Colatex has a lidocaine analog, right here, which helps it localize in the liver. MyoView, on the other hand, shown to the right, is used to image blood flow in the heart. Lastly, bone tumors and metastases can be visualized using technetium-99m methyl diphosphonate. A proposed structure of this imaging agent is shown to the right here, but the structure is not well known, and this imaging agent likely exists as a mixture of complexes and clusters in solution.
This concludes today's lecture. If anything was unclear, please feel free to ask questions during office hours or try rereading the textbook to clarify concepts. And as always, there are some useful websites and resources in the links below to help with understanding some of the concepts that were presented today.